welcome to our Advanced Therapeutics Webinar series, where we focus on the rapidly evolving field that is advanced therapeutics, as it presents innovative and potentially transformational treatments for children now and in future years. Before we start, I'd like to begin by acknowledging the traditional owners of the various lands on which we are all meeting today. This webinar is being hosted from the traditional lands of the Gadigal and Bidjigal people of the Eora Nation in the east and the Barramadigal people of the Darug Nation in the west of Sydney. I'd like to pay my respects to elders past and present and acknowledge the community members, Aboriginal staff, services and organisations who work closely with us to improve the health and well-being of children and young people, their families and communities. So today we are joined by a large panel of experts from the network as we revisit the topic of ocular gene therapy. I'd really like to welcome Robin Jamieson, Benjamin Nash, John Grigg, Brad Rockcliffe, Emily Chin, Matthew Simonovic and Alan Ma, who will take us through the various aspects of the introduction of LUXTURNA as a life-changing therapy for an inherited blinding eye condition. Following their presentations, our panel will open for discussion, so please submit any questions throughout the webinar via the Q&A tab in the Zoom controls at the bottom of your screen. And if you see a question submitted that you'd also like to have answered, please make sure you upvote the question by clicking the thumbs up beside it. Now I'd like to hand over to Robin, who is the lead of Oculogene and Cell Therapies Australia, to begin with the introduction to LUXTURNA gene therapy. Thank you, Robin.
Thank you very much, Paula. I'll just share my screen. Hello, can everyone see that?
Looks good, Robin.
Thank you. So thanks and welcome everyone. So we want to talk today about clinical LUXTURNA ocular gene therapy and vision rescue and our health system team approach. Because this has been a very large effort from many teams across our institutions. First of all, we're having Benjamin Nash from Sydney Genome Diagnostics, John Grigg, an inherited retinal disease specialist, Brad Rockcliffe from the pharmacy, Emily Chin representing nursing and operating theatres, Matt Simonovic from vitreoretinal surgery, and then Alan Ma looking at health implantation research to see how we can implement this work in the most affordable and appropriate way. So in our approach in ocular and gene and cell therapies, we are very interested in impaired vision, particularly due to retinal and macular diseases, inherited retinal and macular diseases, and taking a genomics approach to genetic diagnosis and novel disease gene identifications. And then if we find that a gene is suitable for a therapy approach or a clinical trial, then we want to take that to the patient as quickly as possible. In some cases, as you'll see from the presentation, functional studies are required to facilitate that. And so we proceed with those to again take the patient through to treatment as we are able. And also there is a research arm to this as well to develop novel therapies. But then, of course, as well, we need the health system implementation aspects to get this to the people that need it in as equitable and efficient manner as possible. So to make retinal gene therapies equitable and accessible, then we need to be very aware that these conditions, the inherited retinal dystrophies, do have a lot of clinical and genetic heterogeneity. So that requires detailed ophthalmic assessment as well as a genetic and a genomic approach to find the genetic cause. And since we're talking about gene therapy, it's not possible, generally speaking, to give a specific gene therapy without actually being able to know the genetic cause. And then, of course, the eye is a small organ. It has accessibility, so it is an ideal place to start with gene therapy approaches. So we find that the ocular genomics multidisciplinary team meeting is a particularly useful avenue to start off with our patients to understand their phenotyping, the genetic aspects, and then marrying up the phenotype and the genomic diagnosis, dealing with variants of uncertain significance, so that we can find the answers from a genetic perspective from these patients, and then consider, does the genotype fit the phenotype? Segregation studies are appropriate, including phenotypic review of the family members, and then we need to consider clinical ophthalmic staging. We'll take you through these various aspects with the experts in these areas through this presentation. And then RPE65, which we're particularly discussing in this presentation, particularly goes along with Leber congenital amaurosis and early onset severe retinal dystrophy, and sometimes also the patient might present with a diagnosis of retinitis pigmentosa. So then ocular gene therapy, it's been a long road. There were in the 2000s animal model studies, and then a period of around at least 10 years of clinical trials for this first adeno-associated virus delivered gene therapy for inherited retinal diseases that's become clinically available in Australia. In 2017, there was FDA approval for this gene therapy. This was followed in Europe, and then in 2020, we had TGA approval in Australia, and then 2021, MSAC approval, so there is federal and state funding now for this therapy. So the TGA has particular criteria which are required to be met for RPE65 gene therapy. So the clinical diagnosis needs to be appropriate with early onset retinal dystrophy or Leber congenital amaurosis, most usually, with typical clinical features, some of which are seen here with the appearance in the fundus, the retinal flecking, which is sometimes seen, and then the loss of autofluorescence, which is sometimes seen on the wide field autofluorescence. Genetic diagnosis has to show pathological biallelic RPE65 mutations, and Ben will talk further about that. And then sufficient viable retinal cells have to be present with a clinical assessment, and John will talk further about those aspects. So we established a 16-member working group in August 2019 because we're aware that we had patients with suitable mutations in this gene, and so we needed to have the genetic aspects sorted, the ophthalmic assessment sorted. We worked towards, with others, the TGA approval, and required Office of Gene Technology Regulator and IBC committee approvals because of this AAV therapy. And we needed to consider the pharmacy requirements and the nursing and surgical requirements that we're going to talk about today. The operating theatre requirements, and Matthew will talk about those further, and then all of the ophthalmic equipment required, the workforce development and training, and the standard operating procedures, and funding arrangements, of course, were critical. And then IRD genomic testing and NDT needs to be continuing for service sustainability. So as you can see, this is quite a large team of people involved in this, and this doesn't include everyone. However, with this setup, we've been able to determine that 5 patients have eligibility for this TGA-approved therapy, and those patients have been successfully treated with follow-up investigations proceeding. And so today we're talking about the team contributions and the health system sustainability approaches that we're taking. So thank you, and I'll pass over to Ben to continue our presentation.
Thanks for that, Robin. And I'd also like to thank the organisers for the opportunity to present at this meeting, and to begin by saying that this is actually a collaborative approach and a collaborative work which I'm talking about today between Sydney Genome Diagnostics and the Genetics Research Unit of the Children's Medical Research Institute. So I guess today I'm really going to talk about the genomics and the genomic eligibility for LUXTURNA therapy. And to do that, I thought I'd discuss a case. So this case I'd like to present today was a proband who presented for ophthalmic investigation at 4 years of age. And they did have a flecked macular appearance on wide field fundal examination. So full field electroretinogram, which is displayed here in these traces, showed extinguished scotopic responses at 5 years of age. And you can see that in these top figures, sibling 1 and sibling 2, compared to the normal traces below. On further repeat examination at 12 years of age, this was quite an extinguished trace. So looking at optical coherence tomography, or OCT, there was noted to be a preserved photoreceptor layer within the macula, indicated by this orange arrow here. And this led to an ophthalmic diagnosis of early onset severe retinal dystrophy for this family. So then looking at genomic studies to try and determine the genetic cause of this presentation, commercial targeted capture gene panel testing was undertaken, and this targeted capture was looking at examining 280 inherited retinal dystrophy disease genes. And this study identified compound heterozygous changes, so 2 heterozygous alleles on different chromosomes in the gene RPE65. So the first variant was a missense variant at amino acid position 91, and this was classified as a pathogenic change. This is the most commonly reported pathogenic variant in the literature. It's been reported in many individuals with Leber's congenital amaurosis, early onset retinal diseases, and retinitis pigmentosa. And subsequently, there have been experimental studies which have shown that this missense change in particular results in normal mRNA expression, but then a rapid protein degradation in cell culture of RPE cells. The second variant which was identified was a synonymous substitution, meaning that the DNA aberration did not result in a change in amino acid. And this was at position 31. And subsequently, this was classified as a variant of uncertain significance on this clinical reporting. So this was a novel substitution. It was absent from our control genome databases, such as NOMAD, and also from disease variant repositories, such as ClinVar, LOVD, and HGMD. And this particular variant indicated in red here on the gene structure diagram, we can see that it is quite close to the intron-exon boundary of exon 2, and actually it impacted the penultimate 3' nucleotide of this exon. Looking at our in silico or computer prediction software, we could see that this change at the DNA level was predicted to reduce the effect of natural splicing at the canonical donor site. Over on the right, I put a figure here of the RPE65's location in the visual cycle, and you can see here that it is actually an isomerase or an enzyme which is responsible for the conversion of all transretinal ester into 11-cis retinol. And you can see and appreciate that if this enzyme is not functioning correctly, we have a failure in the visual cycle of these affected individuals. So looking then at LUXTURNA eligibility and genomic diagnosis, one of the TGA criteria is that you need to have biallelic pathological variants in RPE65. And further to this, it should be confirmed by a NATA-associated testing authority accredited laboratory, or a NATA accredited laboratory, which at the Children's Hospital of Westmead, we are a NATA accredited laboratory. But as you can appreciate, we have one variant which is of pathogenic effect in this particular example family, and one variant which is of uncertain significance. So at the moment, this family would not be eligible. So subsequent studies would need to be undertaken. So to do this, we sought to apply a stem cell-based approach to model this variant of uncertain significance. So we had access to a parental carrier's peripheral blood mononucleated cells, and we were able to nucleoffect these with different transcription factors to induce pluripotency. These cells were then manually isolated and expanded, and ultimately, after multiple rounds of cell culture, were isolated into retinal pigmented epithelial cells, which are the cells which express RPE65. So these cells were characterized with trilineage differentiation, SNP arrays to determine the genomic stability after multiple rounds of cell culture, and also then RPE marker expression using immunohistochemistry to ensure that we did have the correct cell type, which would express RPE65 in this carrier. We then undertook cDNA studies looking at extracted RNA from these cells. And you can see that using multiple primers, we were able to amplify this particular region. And then on our agarose gel, we can see that in the parental carrier, we have 2 bands. So one band representing the wild type or the correct splicing for this region, and then the smaller band, which represents exon skipping of exon 2 due to this variant. and which isn't seen in our control equivalent RPE cells. So running these bands then through Sanger sequencing, we can further determine that and illustrate that exon 2 has in fact been spliced out. And this would then result in a truncated protein of only 22 amino acids, and thus be classified as pathogenic. So this result was then discussed at our Oculogene multidisciplinary team meetings, which are virtual meetings held monthly, bringing together clinical genetics, ophthalmology, and the laboratory genomics components, where we were able to discuss in this case and its suitability for LUXTURNA therapy. And as such, this result and variant was reclassified to pathogenic, then confirming eligibility for this patient. So with that, I'd like to thank everyone and pass on to John, who is going to discuss the ophthalmic and clinical eligibility requirements. Thank you.
Thanks very much, Ben. So in my section, I want to sort of discuss the approach we have to people with suspected inherited retinal diseases, and then how we monitor them and their response to therapy. And in a broader sense, we can split our assessments into structure and function. So functional tests, so many of you are familiar with visual acuity and color vision. And then visual field testing is sort of brought into two broad groups. There's the sort of sensitivity, where we change the intensity of the light, and then there's kinetic perimetry, which measures the boundaries of that. And then and that's an important one that we use for assessing how much people have in their visual field. Then visual electrophysiology, and a new test we'll talk a little bit about is full field stimulus test, because one of the challenges in assessing function in these people is if you've got very poor vision, how can you measure change? And that's been one of the challenges for developing clinical endpoints, particularly for therapeutic trials, but in also monitoring response to treatment. And then finally, the other area of structure, where we can take the regular photographs that people have seen. Fundus autofluorescence is a relatively new technique which looks at particular properties in the retina and how they fluoresce. And certainly in RPE65, because of the lack of the enzyme, then you don't you have a gap, and so you don't get the normal compounds that will fluoresce. And we've seen some photos of that already. And then we can have the cross-sectional studies, which we see with optical coherence tomography. So when we look at patients as whether they might be suitable for LUXTURNA, for retinal gene therapy, we have to see whether there's sufficient viable retinal cells. One of the things about patients with RPE65 disease is that there is a mismatch between structure and function. And by that, we mean that they have quite profound vision impairment. However, they still have preserved retinal function. And that's sort of probably been one of the keys to success in the therapy of this, is that there's still structural elements present, which then can be targeted with therapy, but they've had profound visual loss. So they've had, they've been blind since birth, but they have the structure for which the therapy can work on. And so in assessing viable retinal cells, it's not just one measure, it's looking at the constellation of all these factors of visual acuity, visual fields, OCT assessment, the full field sensitivity test, and mobility. And they don't get equal weighting. We then weigh up on how that impacts on the patient's overall function to determine whether there's viable cells. So each of those components contribute, but there's not magic cutoff numbers at all. So at the top here, we can see the kinetic perimetry, and you can see a blue and a red line, and they mark the boundaries of the person's peripheral field. So you can see, particularly say the red target, it's about half the size of the normal red band you can see on the printout there. So that's a reduced peripheral field. So this is one of our younger patients who went forward. I think they were the second patient having therapy. You can see on their cross section that where the red arrow points, that points to the ellipsoid zone, which represents part of the outer retinal structures where the photoreceptors are, the rods and cones, and that layer is still intact. So we can see that there's structure there and there is still some function. This is the patient again, and the bottom shows after therapy. So there's been an increase in their visual field, and also we can see that there has been some increase in the visual acuity, which has not been always the case with this therapy. So in the clinical trials, there's no statistical improvement in visual acuity, but we've seen it certainly in our young patients. They have had improvement in their central vision, and which has been nice for the patients, but not completely expected. One of the tests was particularly developed for assessing people with extremely poor vision is called the full field stimulus test. Literature has a couple of other names, but the full field stimulus test is a measure of sensitivity of the entire visual field, and it estimates the lowest luminance or the lowest stimulus of a flash that elicits a visual sensation. And so we're really looking for little pockets or islands of retina that are still having some function. And this is important when we have these severe vision impairments, such as with Leber's congenital amaurosis. It gives you an indication that there's still functioning retina present, and it's a way of being able to measure people's visual function over time. This is particularly important since most of these people with Leber's congenital amaurosis have difficulty seeing the top letter on a chart, or can only see count fingers or hand movements, or they have difficulty with their visual field tests. This provides a level of retinal sensitivity, but it doesn't provide any spatial information. It's quite an involved test. It takes 40 minutes of dark adaptation. So the patient has to sit in a completely dark room for 40 minutes, and then they have a series of lights presented of varying intensity so that we can find the boundary where they can see and they can't see. So that's a practical impact for assessing these patients. It's really a half-day testing by the time they go through this. So here's the second patient I wanted to talk about. So this is a patient who had visual problems from around the age of 2 to 3. That's when they were first recognized to have difficulty with night vision. And that's one of the historical clues with people who might have RPE65 related retinopathy, in that they have quite profound night blindness or nyctalopia from a very early age. It's unusual for children to complain to parents that they can't see at night, but this cohort will actually start to do that when they become verbal around 2, 3, or 4. This patient had 6 siblings and 2 were affected. And these are the results on the left from when he was 36 years of age. And you can sort of see on the top here, these yellow flecks. This is a more traditional fundus camera view, but it's only a narrow view. Then on the wide field imaging, we can see some increased autofluorescence in this area, and then we can sort of see that there's still the outer retinal structures there. By 2021, we've now got a wide field camera, which can show there's a couple of pigmentary changes. So sometimes this could be misdiagnosed as retinitis pigmentosa. Then here, now we can't see any of the blood vessels at all. So there's a lack of autofluorescence here because the blood vessels normally don't autofluoresce, and this time the retina is not fluorescing. So it's all just one uniform sort of. When the technicians take this, they think they've got an error in taking the scan, but that's one of the clues that this might be significant. And here we have their outer retinal structures, and again, we still have preservation of the outer retinal structure, so we know there's photoreceptors present there. And this person here, if we look on the right, you can see how tiny their visual field. So there, by the time therapy came, this person was in their late 40s, so there wasn't really a lot of fields. They were really constricted visual fields and tunnel vision approaches. But our FST here, you can see that the line, so a shift in retinal sensitivity means the curve moves from the center to the left, and we can see that here from the pre and post therapy. So there's quite a shift of 2 log units or about 20 decibels. So really a big improvement. But what that might look like good numbers for the clinical trial, but what that meant to the patient was that this person who was really struggled to use public transport, they could actually get onto a train and find a seat without sitting on somebody else. They could now walk down a street, a footpath at night, and know that there was cars there. They really didn't rely on their cane as much. And our younger patients have been able to see stars in the sky and leaves and things in the trees at night, and have often stopped using their canes. So that's sort of the personal or patient-reported outcomes which match these sort of more technical assessments. So that's the conclusion of my section, and I'd like to hand on to our colleagues for the pharmacy discussion with Brad Rockcliffe. So Brad, thank you.
Thank you for that, John. Just share my screen. Okay, so my role in the ocular gene therapy team is to manufacture and provide the diluted LUXTURNA to theatres for the operation. So I'm going to briefly talk about a bit of a wider scope thing of general pharmacy department requirements for providing LUXTURNA gene therapy. So here I've outlined some of the things that we as an institution and a pharmacy department have to try and consider before we kind of delve into the world of gene therapy manufacture and manipulation. I think these are good starting points for any pharmacy department that wants to kind of, or that needs to look into this pathway. So firstly, are your staff appropriately trained? I'll go into that a little bit down the line, but it kind of goes into the next section of how are these medications going to be used? Are they going to be attached to your hospital's formulary? Do they need certain drug committee approvals? Can your staff make this medication safely? And can it be safely given to the patient? How is this medication going to be prescribed? So novel therapies like this often provide a challenge for pharmacy departments and prescribers alike, trying to figure out the best way to chart them in electronic medication systems, as well as dispensing them in our iPharmacy system. Is there a process for stock management and ordering? How are you going to provide a record of manufacture? All of these things within these electronic medication systems need to be thought about prior. And does your institution have the required procedures and policies in place now to be able to do this? Or is it something you're going to have to work towards? So you can see on the right some of the policies that we've had to look at and put in place. Things like use of dry ice, administration and handling, as well as transport and spill policies. So the process of ordering LUXTURNA is quite complex, and that's largely due to the MDT that's responsible for the administration of LUXTURNA, as well as the cost of the therapy itself. So because of the way that LUXTURNA is, gene therapy is in general, it's ordered per patient, specifically after the medical team have triaged and approved it, and after any other institutional committees or approvals have been signed off. Our chief executive signs off on the invoice themselves, and that's essentially due to the fact that it is so expensive. It comes in at nearly $500,000. So it's a very expensive therapy. The next part that the pharmacy department kind of has to consider is the custody. So once you order it, you kind of got to make sure that you've got the system in place to get it here, and to get it here in a usable way. So we constantly communicate with both Novartis and whichever courier is shipping it to us, making sure we know when it's going to arrive, where in the hospital it's going to arrive, make sure we meet them wherever it is. And then as soon as it gets here, we start the process of QA. So running through, making sure that all of the temperature monitoring tags have been included, downloading all of that data, ensuring there's been no excursions, temperature excursions, and visually inspecting the stock. If there's something wrong with it, we need to know about it right then and there, because it's LUXTURNA and the process of administering it is quite time sensitive. The other thing to consider is it has to be securely stored in a monitored -80-degree freezer, which is a big factor for a lot of institutions because most places don't just have a -80-degree freezer lying around. So training. From a pharmacy side, there's essentially two sections of training. If you break them down, it looks at, is it safe for your staff to make this product? And when you make it, is it then safe to give to the patient? So firstly, any pharmacist or pharmacy technician that's involved in the manufacture is required to be aseptically validated. So this validation is performed by the institution, usually at a departmental level. So we perform our own aseptic validations in-house. And that's just to make sure that our technicians and pharmacists know how to cleanly scrub in and out, how to use laminar flow, biohazard hoods, and sterile manufacturing techniques. Most other hospitals and other institutions will have similar processes. And the other one is biosafety training. So it's required by all staff who are involved in any form of ATMP handling. And that goes through things like what to do, biological spills, how to handle these chemicals safely, therapeutic handling, waste monitoring, and how to operate inside of a specific PC2 facility. So triaging the patient. This process is not too different to how a pharmacist would assess the appropriateness of any medication. It just seems a little bit more complex due to the nature of the medication. But the process is that we would ensure that any approvals through our institution are there before we do anything. Make sure everything's approved before we even start. The next one is we check the medical record, check their medication history, and make sure that it is appropriate. One of the big things we look at are vaccination status. So these patients do end up with a course of systemic corticosteroids. So it's important for things like, are they due any live vaccines in the near future, at which point we would recommend delaying those. We would verify, dispense the medication, do all the labels as legally required, counsel the patient. This is primarily on the corticosteroid treatment, not so much the gene therapy itself. Manufacture and prepare it, which I'll get into next, and then provide the dose. So dose preparation, so this is the fun part. So create the labels prior, make sure we've got all that before the day. LUXTURNA is a very time-sensitive process, involves constant communication between the pharmacy department and the operating suite. So we don't want to go looking for random labels the morning of. We have to defrost the product, so it's -80, so it does take about 30 minutes to defrost. And something that is quite unique to LUXTURNA, so anyone that has any aseptic manufacturer experience, is that LUXTURNA takes 2 operators to be inside the biohazard hood at the same time to manufacture the product and make sure it is completely aseptic. We then provide 2 syringes, 2 theatres of 0.8 mils. A dose is 0.3 mils. And so we draw these syringes up, deliberately overfilled beyond the actual administered dose to account for the extension tubing, as well as any dead space in the syringe or the subretinal cannula. We make 2 syringes just in case something happens to the primary syringe. We only have 4 hours after we defrost the vial, so we really need to make sure we get everything done in a nice, timely manner, and that if anything does happen, we can fix that. So this is just a quick look at the corticosteroid plan I was speaking about. So on the left here, you can see the PI's recommendations for the steroid treatment. Essentially, it's about 1 milligram per kilo, once daily for 3 days prior, and then 4 days post, and then starts a prednisolone wean. On the right-hand side, you can see these are our in-house corticosteroid weaning plans, and this is what we use to counsel the patient and the family prior to surgery. Thank you very much. I'll now pass over to Emily, who's our next presenter, and she'll talk about the nursing and operating theatre requirements.
So my name is Emily and I am a theatre nurse, so scrub scout at Children's Hospital at Westmead. So before we train any staff member to even do an eye surgery with LUXTURNA, you actually need to train staff members to go through a biosafety certification to even also be in the room as well. So these staff members, including myself, we undergo this biosafety training because it actually equips us with the skills necessary to handle infectious organisms and biohazards, which is what LUXTURNA is. We follow a policy and procedure in the hospital, and this document, what it does is that it outlines the procedure followed for transport, management of waste and management of spills, and anything that relates to GMOs across the Sydney Children's Hospital network. So this policy applies to all staff members, and that includes clinicians, RNs, pharmacists, pharmacy technicians, and clinical research staff. This policy also has a spill management procedure. So if for any reason something happens either along the way or in theatres or during administration, we can at least follow the spill procedure to safely discard the GMO that's been exposed. In terms of PPE and safety, as a scrub scout, we all have our standard PPEs that we have in theatres. So you've got your surgical hat, your appropriate mask, depending on the precautions of the patients, and then any shoe covers if you are wearing outside shoes within the theatre. As a scrub, you don your usual surgical gown, always double glove, and your eye protection and the appropriate mask, depending if your patient has, say for an airborne procedure, you would need an N95. As a scout, we always don our mask and eye protection. And when you're handling anything that is a risk of splash exposure, that was when you would don your gloves. So every time in theatres, when the scout is to open the LUXTURNA syringes, we'll always put gloves on just to protect ourselves. So this is a little room setup that I've drawn for theatre, and we do this in Theatre 7 only because the Theatre 7 is our designated ophthalmology theatre. And the reason we use Theatre 7 only is that if we keep it to one theatre, it creates that consistency. So the flow will be the same, and then the way the room will be set up is the same. And this helps other junior staff members who haven't worked with LUXTURNA at all, how to set up and how the flow goes, especially with when we have visitors come in to watch the procedure. So we ensure that only the core staff members are to be in the theatre. So that includes one anaesthetist, 2 nurses, which is one's a scrub, one's a scout, and 2 surgeons. So you've got a lead surgeon and you've got an assistant surgeon as well. The reason we limit staff members in the room is because our Theatre 7 room is actually quite small. So we want to reduce the work health safety hazard. And when you're working with a GMO, you also want to limit the amount of exposure to a minimum as well with the staff members. So outlined in red here is typically the anaesthetist area. And then outlined in blue is where most of the sterility will happen. So from here, the scrub bay, whoever's scrubbed up will walk in through here, and this is where the scrub scout area will be, and this is where the surgeons will be with the 2 smiley faces. Here we've got our clinical waste area, as it's a biohazard. We have a yellow clinical waste bin inside the room, and outside the door is an incineration bin, which I'll talk further on. So in terms of procedural overview, once the patient is intubated, the leading surgeon will contact pharmacy, and then pharmacy will start preparing the syringes. From then on, like Brad said, we only have a 4-hour administration window. After that, the pharmacy will come to theatres and they'll escort a esky and a spill kit. Inside the esky, there will be 2 syringes. One is a primary and one is a backup. We always open the primary first, and the backup is there for the same reason as a backup. Once the LUXTURNA has been delivered into theatres, the surgeon will dilate the eyes and then the surgery will begin. So this is just a picture of our microscope that we have currently in theatres. It is our OCT microscope, and we only have one in the facility. We also have a policy on how this gets transported from our equipment room to theatres, that it's a 2-person job. It is very heavy and very expensive. This is a little photo of what the surgery would look like. So surgeons will do a 3-port pars plana vitrectomy, and then they would do a posterior vitrectomy, and then they make a small bleb with BSS first to confirm the position of the LUXTURNA. Once they confirm the positioning with the OCT, then they will inject the real LUXTURNA. This is our waste management and non-disposable equipment protocol. So products that involve GMOs or potentially contaminated by GMO material, they're categorized as a biohazard waste. So what this means is that waste generated during LUXTURNA, that could include, your disposable PPE, needles, dosing syringes, giving sets, and extension sets. They all have to go into a yellow biohazard bag. So anything that is thrown into a yellow biohazard bag must be disposed of in this yellow bin. And you can see up here, there's a little lock on it. Our CNS2 has the key. Once everything from the theatre has been disposed of, you must put a must incinerate sticker on the lid. The CNS2 will lock it, and then we actually give it to Kids Research to be incinerated by an external contracted company. For our sharps as well, we will have a little sharps vessel and put all your sharps in there, close the lid, and that also goes in the yellow lockable bin. In terms of equipment and sterilization, first of all, we denature the backup syringe that we haven't used. So one bottle you'll open it up and you'll squirt the backup syringe into the 1% Virkon. And the Virkon gets poured out into a jug here, as you can see. So anything that's been touched by LUXTURNA, so in terms of instruments, trays, it has to be denatured by this 1% Virkon. So Virkon is a multi-purpose disinfectant. So it gets soaked in for 30 minutes. After 30 minutes, we send it off to CSSD, and CSSD continues our typical cleaning process and sterilization. At the end of the procedure, in terms of room cleaning, if there's no spill, you just do a standard room clean. If there has been a spill, you follow the spill protocol, and then you use it's a terminal room clean with Claw Clean tablets, which is like bleach. And that's it. So I'll pass on to Matthew Simonovich, who will be talking about the surgical delivery of LUXTURNA.
Thanks very much, Emily. So I'll be talking about surgical considerations in LUXTURNA gene therapy. I'm a vitreoretinal surgeon at the Kids Hospitals as well as at Sydney Eye Hospital. The surgery for LUXTURNA, as we've heard, involves an injection underneath the retina into what is called the subretinal space. So this is what is known surgically as a potential space, in that there's usually, in fact, no space there, just two adjacent tissue planes, that is the neural retina and a supporting layer, the retinal pigment epithelium. So you need to open up this space to deliver LUXTURNA. As we heard, surgery first commences with a procedure called a pars plana vitrectomy. This is a type of micro keyhole operation in which we make 0.5 millimeter diameter incisions into the white of the eye. Then we use sort of small instruments, the size of small needles, to remove the gel that fills the inside of the eye, the vitreous it's called. And following this, we get access to the retina, and it's now that we perform the subretinal injection. And this is the most delicate part of the operation. I've got a diagram on the right of a subretinal injection. We've seen a few of those already. And this is because in the patients that we treat, the retina is typically very thin, sometimes only close to 1/10 of a millimeter. If we don't inject deeply enough, the LUXTURNA will end up in the vitreous cavity, the rear cavity of the eye. And this means that it won't have any treatment efficacy. And at the same time, it may promote a lot of inflammation inside the eye. Equally, if we inject too deeply, we may inject into blood vessels or around blood vessels, and indeed we may puncture them, causing bleeding as well as a lack of treatment effect. So taking it a step back, what are the considerations in surgery? Well, once we've selected patients for surgery, which we've heard about already, the patients are placed on immunosuppression in the form of prednisone, and this commences 3 days prior to surgery. The duration depends a little bit on the spacing of treatment between the two eyes, and it's a minimum of 18 days and a general maximum of 30 days, although this can vary depending on whether or not the patients have inflammation following the procedure. LUXTURNA, as we've heard, is stored frozen, and before we give the pharmacy the green light to thaw the drug, we need to sort of check that we have all the equipment and the staff present. And once the anaesthetists are happy, then we can proceed with general anaesthesia for the surgery, and we notify the pharmacy then, who then thaw the drug. And it's taken between about sort of 45 minutes and an hour between us giving the call and being able to receive the LUXTURNA in theatres. As we've heard, 2 syringes are delivered to theatres, a backup syringe as well as the actual syringe. We've already heard from Emily that there's a large team involved in theatres, and each team member has an important role. The surgical team consists of a lead surgeon and an assistant. The assistant helps deliver the LUXTURNA via a manually operated syringe, which is held by the surgeon. I've been performing the surgery with a colleague, Dr. Gaurav Bhardwaj. We take turns to be the lead surgeon. The nursing team look after all of the surgical equipment essentially, and they ensure the smooth running of the case. The anaesthetist and the anaesthetic nurse look after the general anaesthesia as well as general medical considerations for our patients. And once surgery is completed, the recovery staff care for the patients in recovery, and they make sure that the post-operative instructions are adhered to. We've got a photo on the right here of the first case that we did with a cast of thousands watching, and everybody's in the positions that were outlined by Emily in the previous talk. So the aim of surgery itself is to deliver, as I said, LUXTURNA into the subretinal space. It's only a small dose in terms of treatment volume, that is 0.3 of a milliliter, which, as I said earlier on, is delivered into the subretinal space. The initial portion of surgery is similar to any other vitrectomy. I've got vitrectomy playing top right there of one of the older patients we've treated and have performed LUXTURNA subretinal injection on. And their case highlights some of the difficulties that we see in older patients. So he has a significant cataract, and this impedes our surgical view. In younger patients, such as the one at the bottom, the issue there is not so much getting a clear surgical view, but removing the vitreous gel, which is quite firm and firmly adherent to the retinal surface. So that's the main challenge in younger patients. There are 2 competing approaches to performing subretinal injections for gene therapy. The first one was the approach that was used in the clinical trials of LUXTURNA, and it's called a one-step approach. And in the one-step approach, the LUXTURNA is used to define the surgical plane. In the 2-step approach, what we do is we first of all define the surgical plane with an inert solution called balanced salt solution. We are then able to confirm with on-table OCT, which you can see on the top right here. I've frozen the video. That confirms in histological detail, so like we're able to take a slice through the retina, what's going on. And so once we've confirmed that we've actually located the correct anatomical plane, we can then inject into this bubble or this bleb the LUXTURNA solution, safe in the knowledge that it's reached the correct tissue plane. The top video here kind of reiterates the 2-step approach, first of all with balanced salt solution and then with LUXTURNA. How much drug actually gets into the subretinal space? We know that drug is refluxed. You can tell this using on-table OCT qualitatively. Animal models suggest that the reflux of drug is significant, at least 12% or more. Recently, my lab developed a technique to accurately quantify how much drug gets refluxed by staining, in this instance, TPA or tissue plasminogen activator, which is a clot-busting drug, which we inject in patients who have submacular hemorrhage to trace what happens to this drug after we inject it into the subretinal space. And we can estimate by collecting the fluid at the end of surgery how much of the fluorescent material has been sort of extruded back into the vitreous cavity. And it appears that the animal models perhaps overestimate how much drug is typically refluxed into the vitreous cavity. So it appears to be less than 12% in all the patients in whom we've measured it. In terms of surgery to date, how have the patients fared? Well, we've treated 5 patients in total, both of their eyes, so 10 eyes. As I mentioned earlier, different patients present different surgical challenges. However, surgery has been successfully completed in all patients. 2 of our patients developed modest rises in the intraocular pressure following surgery, which was managed with drops. Another patient developed some inflammation after tapering of their systemic steroids, and we managed this through reintroduction of systemic steroids with a slower taper, and this resulted in regression of this inflammation, and they've done well subsequently. John has presented on FST, so the patients, 4 of the 5 patients have already undergone FST testing. All of them have improved, and really the results mirror the results that were seen in the phase 3 clinical trials. And, you know, this is translated into, you know, patient-reported improvements in their visual function. For example, patients noting that they can, you know, see at nighttime as well as their family, or other members of the family who have, quote, normal vision. They can now see stars that they can not recall having ever been able to see. We plan to monitor outcomes in our patients using a new registry that a few of us have been involved in designing together with colleagues internationally. It's called Fight Inherited Retinal Blindness. Thank you very much. I'll hand over to Dr. Ma now.
Thanks, Matthew. I'll just start sharing my slides. So I'll be speaking a bit about the implementation science research approaches to all of this. And you can really see from the last 6 talks that precision medicine has well and truly arrived. We're starting to get precision diagnostics for our patients with accurate genomic diagnosis. And that's then led to a therapy, such as this publicly funded LUXTURNA therapy, which is offering a lot of promise. And so this has then paved the way to a precision medicine approach that holds a lot of promise, not just in ocular genomics, but also to healthcare in general. However, you can also see there are some massive barriers, both on a local level and also systemic level. In the diagnostic space, there really is an issue of limited access and equity issues for diagnostic genomics. In the therapy space, this has all happened so quickly and developed so rapidly that our healthcare systems don't have a systems approach or a model for precision therapies and trials. And so the real challenge moving ahead is, how do we translate all of this amazing work in precision medicine into an actually realized, implemented and translated outcome to improve patient care? In addition to this, you can see all of this work is just in one space, the ocular space, but this is being replicated in many areas: neuromuscular, cancer, respiratory, many areas of medicine. And so this has altogether created a precision medicine tsunami, where there's so much demand for this type of approach. However, limited supply, especially in the subspecialty groups, in the molecular and clinical genetics area, and even trickling down into the system in terms of pharmacy and operating theatre and staffing. There really is a bit of a bottleneck here. In the ocular space, what we've done is really have a team approach, because this is such a complex area, and our interdisciplinary collaborations has really helped in terms of delivering good outcomes. In terms of the Oculogene MDT model, I've kind of split it up into 3 steps here. You have the pre-test type of advice model, where we address a lot of queries from all over Australia about genomic and diagnostic testing in inherited retinal dystrophies. We also advise a lot on testing itself in terms of careful interpretation of genomic results, giving expert lab opinions, and also very careful clinical correlation of complex results. And then after the testing has been performed, we also help advise on clinical trials availability, therapy access, as well as further advice and the logistics as well. And really, we think that this model, where we get together the ophthalmologist, the lab, the research arms, the genetics, counselling and clinical services, pharmacy, allied health and nursing, all together to deliver this outcome helps to improve collaboration, improve teamwork, education, and facilitates not just genetic answers, but management and precision therapy access for our patients. But I guess the question that I've been trying to tackle in my research is to really look at this as a model. Does this model actually work, and is it a good model for future precision medicine approaches? Because as I mentioned before, this tsunami is coming, and this type of pathway to precision medicine might provide a pathway for other areas that are being developed. And so the big question that I'm trying to tackle is, how can this type of model inform system level change and provide value for our patients, for our healthcare providers and healthcare systems, as this demand is inevitably going to increase? And so that's where implementation science comes into play. Now, there's a lot to describe in implementation science, and so I've just tried to summarize it in one slide here. But essentially what it is it's a scientific study of methods that promote systematic uptake of research findings and other evidence-based practices into routine practice. And really the hypothesis that it's based upon is that when we establish that something is clinically effective, such as a therapy or a type of process, this doesn't necessarily guarantee actual uptake into routine usage. And we all know about the practice, the evidence to practice gap of up to 1 or 2 decades between when something is proven to be evidence-based to actually being used to benefit patients. And we can't afford to have such a gap in precision medicine. And so the aim of this research is to identify reasons why certain practices work and find strategies to work out how they can work in a feasible, adaptable, sustainable, and cost-effective way. And so it moves beyond just the traditional does it work type questions of effectiveness studies to how can we adopt, implement, and sustain change in a model in a health system. And so the real aim of this type of implementation for precision medicine is to provide precision diagnostics that not just improve diagnoses, but also improve access and equity for our patients, to provide therapies that help with collaboration and management options for all clinicians, and ultimately a system for precision medicine that's sustainable and adaptable into the future. And so right at the beginning of these talks, you would have seen this slide from Robin, and I guess what we're trying to work on is you can see that there's an incredible amount of work that's been done in the clinical space with our ophthalmology colleagues, in the genomic space for diagnostics, as well as in the functional studies and therapy models, looking at variants of uncertain significance, complex results, as well as stem cells, gene editing, and pharmacological approaches. And all of this brings back to the patient the possibility of new treatments and clinical trials and the promise of precision medicine. And what we're trying to see is whether this particular model may be useful as a health systems approach to be implemented and facilitate equitable access and sustainability and better patient outcomes in ocular diseases, but also in other areas of medicine. I think I'll finish there and I'll hand back to Paula for questions.
Thanks so much, Alan. That was fantastic. And I really just want to extend a really warm thank you for those incredible presentations and absolutely an amazing demonstration all the way through. From research to clinical practice. It's really fantastic to see. I'd also like to thank those who have submitted questions. So I'd really like to introduce Dr. Laura Fawcett, who is a respiratory consultant and the Advanced Therapeutics Medical Lead at Sydney Children's Hospital, and Dr. Michelle Lorenzos, who is the Advanced Therapeutics Medical Lead and Paediatric Neurologist from the Children's Hospital at Westmead. And they're kindly going to take us through facilitating the Q&A today. Thanks.
Thanks, Paula. I'm going to start with going back to Robin for this first question. So Michael Stevens has asked that we were informed that LUXTURNA costs $450,000 per treatment and is interested to know how that cost is met.
Thanks very much for the question, Michael. So this treatment is a relatively expensive treatment. It's a one-off treatment. That's it for a lifetime treatment. And this has been analysed and approved through the MSAC process, has had a health economic analysis related to the impact of this on people's lives and their working capacity and so on, and has deemed to be appropriate. And it's now covered by a combination of federal and state government funding, and as we've seen, is providing real benefit to patients.
I might add with that too that it's quite unique that it's $450,000 per eye, and it's one of the only medications that I know of that the drug company Novartis actually provides, or a secondary payment for when clinical criteria have been met. So we pay for the first payment at surgery, and then the second payment is actually made if the patient reaches certain clinical criteria, and that's quite unique.
Thanks, Brad. So I might just go to a question by Callum Jin. Probably a question for Matthew or one of your, or feel free to go to one of your ophthalmology colleagues, but quite a technical question. So is there a specific layer aimed for with the intraretinal injection, or is close enough effective?
Can you hear me okay?
Yes, we can.
Yeah, sorry. So there is a specific layer. As I mentioned earlier on, that layer is a potential space between the neural retina. So that's the part of the retina does the sort of the business of vision, detecting light and transmitting information to the parts of the brain involved in vision, and the layer underneath it which supports it called the retinal pigment epithelium. So that's the layer that you're aiming for, and you know, you've pretty much got to get it into that space. The retina tends to want to detach at that location, so you know, that's working in our favor, but it's pretty precise.
Thanks, Matthew. Next question starts off with a comment to everybody to congratulate you on an amazing team effort. And then they're interested to know what the success of the LUXTURNA means for other ocular conditions and how the same technology could be used for other genetic conditions of the eye. So who wants to take that first?
I'll answer that, and Matt might be interested in answering that as well, and others. So certainly, yeah, this gives us a lot of hope for this type of approach for these retinal conditions. There are a lot of genes, so that's one thing to consider, but it's having good effect in this particular case. There is an enormous amount of work worldwide, other institutions, and as well as our institution here, looking at other genes and how they're suitable for gene therapy using model systems, so using induced pluripotent stem cells from patients and testing novel therapies as well as other model systems for these types of approaches. And also people are considering other more general approaches that might impact particular pathways that are known to be involved in these conditions, which can be a useful approach that can be beneficial for more than one particular type of genetic cause. And Matthew might want to make mention of optogenetic approaches, which are another type of approach that's being investigated.
Yeah, sure. Thanks, Robin. So as Robin said, many genes or loci have been implicated in inherited retinal diseases, more than 300. And so part of the problem of developing treatment is this diversity. And Robin mentioned optogenetic gene therapy. So this is sort of suitable for people who have advanced sight loss. And the common pathway to losing sight in many inherited retinal disorders is loss of the light detecting cells in the retina. But in most of these patients, the cells that normally convey information from the retina to the parts of the brain involved in vision survive, but they're not normally light sensitive. So optogenetics is a type of gene therapy which aims to introduce light sensitive proteins into these relay cells to make them light sensitive, and that restores vision. Or that's the theory at least.
Okay, thank you. So this is a question that I suppose several of you may be able to answer, but how long does the treatment last? So Robin, you mentioned that this is a lifelong treatment. So is there confidence at this stage that it will last and continue to have the same kind of potent effect over that whole lifetime?
There are some results from outcome studies, and John John's been involved in, we're involved in the PERCEIVE study and other studies looking at long-term effects.
So there's been a number of clinical trials that have gone for 15 years now. So there's some patients have gone 15 years with good effect and maintenance of their improvement in vision. I mean, and that's the challenge, how long will it last? There's a very good animal model, the Briard dog, which has gone 17 years. And so in dog years, that sort of could be converted over to 80 or 90, depending if you believe that conversion. So for humans, though, it's been at least 15 years now since the first patients were treated with the LUXTURNA or variations on that. But to try and analyze that going forward, there is a real-world post-therapy study which the Children's Hospital is part of called PERCEIVE, and that's a European, it basically is non-North American because Novartis has the rights to LUXTURNA outside America. Spark Therapeutics, who developed it, have the US rights, and Novartis has been developing this registry, which will go for 10 years.
Thank you, John and Robin. So there's a question coming in from Jing on the topic of inflammation. A question about whether the inflammation will affect the efficacy of LUXTURNA, and that some patients can have subtle subclinical inflammation and the effect of that on long-term effect on vision, and whether it's detectable by any tests.
Well, I could start with that and then Matt could perhaps add to it. So inflammation can be an issue, and that's why regimes of immunosuppression have been developed. And so all patients undergoing this therapy will have a minimum of 18 days of prednisone. And so if it's suppressed, then it doesn't seem to affect efficacy, and that's been the results from the clinical trials and so far with our clinical patients we've treated. If it's significant inflammation, and there are a small number who will have that, then that has potential to impact on efficacy, but by them managing the inflammation, that will reduce that. And I think Matt's had been managing a couple of the patients who've had some inflammation. Matt, if you wanted to comment.
Yeah, sure. So just like John intimated, yes, if you do have active inflammation, just by virtue of things like a pacification of the ocular media, that is the vitreous cavity, it can make vision hazy purely because of inflammation within this part of the eye. Once this is dealt with adequately, then vision will be restored and get back to kind of a treated normal or post-treatment normal, if you will. So, the evidence to date suggests that, active inflammation can be treated successfully, and once it subsides, it doesn't affect efficacy.
Thank you both. We have another question coming in from Madison Wiggins. So for patients who've had genetic testing at their local genetic service, what is the process in accessing the MDT if there's uncertainty regarding variants and or potential for therapy or trials?
That's exactly what our MDT is set up for. So thanks, Madison. And Ben is our contact person. So Ben Nash, who presented second. So Ben, you might want to talk a bit about that.
Thanks, Robin. So generally we hold these meetings monthly, and we try to allocate a 20-minute session for that multidisciplinary team discussion. So we have the ophthalmology team involved, the referring clinical geneticists and genetic counselors who have seen the patients, and we get copies of all the diagnostic genomic reports. So it doesn't necessarily have to have been seen here. These reports sometimes come in from an overseas laboratory and may have been interpreted differently to how we would interpret things here in Australia. So it's good to have those discussions. If there's any particular cases of interest, generally people email me or Robin or any of the team, and we can facilitate a slot in our waiting list and go from there.
Thank you. And then I've got a question here for Alan. It's an anonymous question, but they'd like to know, why do you think we experience so many challenges with the implementation of these precision medicine approaches when they're obviously needed and have demonstrated such obvious success and clinical outcomes?
Oh, what a question. This is what keeps most of us up at night, isn't it? I am literally about to submit a paper that addresses this exact question. So keep your eye out. We'll send it out once it gets, published in the literature. But I think a short answer is, I think it's just a problem of demand and supply, that the demand of this disruptive technology of precision therapy, genomics, over the last 10 years has just exploded exponentially. The supply is probably actually reduced. Our health systems aren't barely coping. COVID, everything has just wiped everything out. And there's 150 geneticists in Australia. We can't diagnose every single patient with RPE65 in Australia, well, in a useful time frame. So the supply is way outweighed by the demand, and so we need to use smart systems to develop to address this, that doesn't just burn out our existing staff, but also is cost effective and sustainable, because this same story is going to repeat again and again and again in the next area of medicine that explodes like this. So I think I'll stop talking there, but I think it's a massive problem.
We have time for one more question, Laura.
Thanks, Paula. I might put there's two very quick questions. I think they're quick questions in the chat left. So a couple of questions about specifics. So does it work for those who are already legally blind and do not have enough live cells? And a question about prednisolone dosing, maximum and minimum, and PPIs. So some specific questions there.
John, you want the first one, yeah.
So legal blindness is a technical definition that's less than 660, and so that's one thing. So legally blind, it's still suitable for therapy, but the critical thing is you have to have enough viable retinal cells, and that is one of the requirements for this. So without enough viable retinal cells, then we won't do the therapy. We go to the prednisone dose. The protocol maxes at 40 milligrams, and that's for about half of those 18 days. So 40 milligrams for 3 days before, 4 days, including the day of surgery afterwards, then tapers, and then the second eye goes back up to 40 for the 3 days before and 40 milligrams for 3 days after, and then it halves for 7 days and halves again.
We wouldn't usually provide a PPI for a short duration either.
No.
Thank you, Mo.
Excellent. Well, I think we are at time, so I really wanted to just say firstly, thank you for joining us today.